This method can help answer key questions in the autophagy field, such as how autophagosome formation is regulated, and how various cell treatments and conditions affect bulk autophagy. The main advantage with this technique is that it measures endogenous cargo sequestration. Therefore, the method is broadly applicable and independent of artificial overexpression or introduction of cargo probes. Demonstrating the procedure will be Paula Szalai and Morten Luhr, PhD students from my laboratory.
To begin, culture adherent cells in 75 square centimeter tissue culture flasks in a humidified incubator with 5% carbon dioxide at 37 degrees Celsius. Allow the cells to grow until they reach a near confluent cell layer. After this, wash the cells with 3 milliliters of PBS at 37 degrees Celsius and pH 7.4. Remove the PBS and replace it with 3 milliliters of 0.25% trypsin EDTA. Incubate in a humidified incubator with 5% carbon dioxide at 37 degrees Celsius until the cells detach. Next, resuspend the cells in 7 milliliters of culture medium containing 10% FBS and transfer to a sterile 50 milliliter tube. Using a 0.5 to 20 microliter pipette tip, transfer 10 microliters of cell suspension to a microcentrifuge tube containing 10 microliters of trypan blue. Use the same pipette tip to immediately fill a counting chamber slide and count the cells in an automated cell counter. Then use culture medium containing 10% FBS to prepare a suitable dilution of the cell suspension.